Welcome students, to today's lecture on the intricate field of amino acid synthesis in the human body. In this comprehensive script, we will delve deep into the biosynthetic pathways and metabolic regulation of various amino acids. Understanding these pathways is crucial for comprehending the underlying physiological processes and the implications they have on human health. Glutamate and glutamine are essential amino acids that play pivotal roles in various biochemical reactions within our bodies. Glutamate is synthesized through the amination of alpha-ketoglutarate by glutamate dehydrogenase or by transamination reactions involving different amino acids. Additionally, glutamate can be derived from oxaloacetate through malate dehydrogenase or directly through the oxidative decarboxylation of alpha-ketoglutarate by the alpha-ketoglutarate dehydrogenase complex. Furthermore, glutamate is converted to glutamine by the enzyme glutamine synthetase. This conversion incorporates ammonia, which is captured in a high-energy bond, yielding glutamine. Serine and glycine are two non-essential amino acids that can be synthesized through the glycolytic pathway. Serine is formed through the conversion of 3-phosphoglycerate by the enzyme phosphoglycerate dehydrogenase. This reaction requires NAD+ as a coenzyme and results in the generation of NAD. Glycine, on the other hand, is derived from serine by the enzyme serine hydroxymethyltransferase. This reaction requires tetrahydrofolate, a cofactor involved in 1-carbon metabolism. Alanine is another non-essential amino acid that can be synthesized from pyruvate. This conversion is facilitated by the enzyme alanine transaminase, which transfers the amino group from glutamate to pyruvate, generating alanine and alpha-ketoglutarate. Aspartate can be synthesized from oxaloacetate through the enzyme aspartate transaminase, which transfers the amino group from glutamate to oxaloacetate. This conversion yields aspartate and alpha-ketoglutarate. On the other hand, asparagine is formed from aspartate by the enzyme asparagine synthetase. This reaction involves the transfer of an ammonia group from glutamine to aspartate, resulting in the formation of asparagine and glutamate. Arginine can be synthesized through two different pathways, namely the citrulline-ornithine cycle and the de novo pathway. In the citrulline-ornithine cycle, ornithine is converted into citrulline by the enzyme ornithine transcarbamylase. In the de novo pathway, arginine is synthesized from glutamate via a series of reactions involving various intermediates, such as acetyl-CoA, acidoacetyl-CoA, and 5-phosphoribosyl 1-pyrophosphate. Proline, a non-essential amino acid, can be synthesized from glutamate through a series of enzymatic reactions. Glutamate is first converted into pyrroline 5-carboxylate by the enzyme pyrroline 5-carboxylate synthase. Subsequently, pyrroline 5-carboxylate is hydrolyzed to form proline. Histidine is an essential amino acid that cannot be synthesized in sufficient quantities by the human body. Therefore, it must be obtained through the diet. Histidine can be biosynthesized through multiple enzymatic reactions, starting from 5-phosphoribosyl 1-pyrophosphate and involving different intermediate molecules. Regulation of histidine synthesis is primarily controlled by feedback inhibition, where the final product, histidine, inhibits the enzymes involved in its own synthesis. Tyrosine is synthesized from phenylalanine through the enzyme phenylalanine hydroxylase. This enzyme catalyzes the hydroxylation of phenylalanine to form tyrosine. This pathway is crucial as it allows for the production of tyrosine, an amino acid that serves as a precursor for important molecules like dopamine, adrenaline, and thyroxine. Cysteine, a non-essential amino acid, can be synthesized through multiple pathways. One of the key pathways involves the conversion of serine to cysteine via cystathionine. This reaction requires the enzyme cystathionine beta synthase and cystathionine gamma lyase. Metabolic regulation of cysteine synthesis involves the feedback inhibition of cystathionine beta synthase by cysteine itself. Taurine is a unique amino acid that is not incorporated into proteins. It can be synthesized from cysteine through a series of reactions involving cysteine dioxygenase and cysteine sulfonate decarboxylase. Taurine has diverse physiological functions, particularly in the central nervous system and cardiovascular system. The liver is a critical organ for the synthesis of non-essential amino acids. It contributes to the synthesis of alanine, glutamine, aspartate, serine, and glycine, which can then be distributed to other tissues for various physiological processes. The synthesis of non-essential amino acids involves complex interactions between different organs. Amino acids are formed in various tissues and transported to the liver for further processing and distribution. Inter-organ cooperation is essential for maintaining amino acid homeostasis in the body. In conclusion, the biosynthesis of amino acids is a complex and tightly regulated process that is vital for sustaining life. Understanding these pathways and their regulation provides insights into normal physiological functioning and the pathogenesis of amino acid-related disorders. Further research in this field holds promise for the development of novel therapeutic interventions and improved treatment options for patients.